we decided to tag team this only because it made sense to show you sort of the temporal flow of the cell generation in Kevin's lab. And I got to tell you, he does a very high-quality job of checking everything very compulsively to make sure they are right. The cells then are now shared with us in Chicago, and we've been analyzing neurons that his lab has made and sent to us. We've also locally generated some neurons and have recorded from them. And our goal is actually now to study them for their electrophysiological properties. Do they fire action potentials? And are there properties of their, are there neurophysiological properties that are distinct in the cells made from the patients with AHC from patients who don't have AHC, what we call normal controls? So anyway, there is another slide after this somewhere. Bill, are you queuing up? He's working on it. To escape out of it, and then we can just reboot it. Anyway, the technique that I'll be illustrating for you is, we call it patch clamping. Doesn't mean anything to you, but this is a method where we take a single cell, we take a small capillary tube, draw it out to make a tiny end, something like 10 microns in diameter, and then that is placed on the membrane of the cell, connection with the cell, and then using some amplification, we can actually record individual electrical events in the cells. And so what I'll be showing you is actually action potentials, the things I started talking to you before. So that's a Chicago skyline. Looks like that every day. This is some pictures of some neurons. There really are three classes of neurons that we have been focused on: excitatory cortical neurons, GABAergic interneurons, although we don't have data for you today. Those were Kevin's favorite cells, as he mentioned. And then lastly, I'll show you just some things we've done with spinal motor neurons. The reason we did that is that it was an easy protocol that was very robust. And so the first thing I'll just show you is this: the timeline of making the neurons. It takes about 3 weeks to make them. They get plated on, or they get seeded onto a dish that has mouse astrocytes, a supporting cell from the brain. And here's some pictures of those neurons. The ones in red are the actual neurons. And here are some action potentials that we recorded. On the left are neurons, are action potentials that were recorded from a person without AHC. Is someone with AHC who has the G947R mutation. These are both female patients or female subjects. And what's not clear, of course, what you're seeing here is there is a time along the bottom, left to right, and then the height of these peaks is really the voltage in the membrane. And so these spikes that go up fast is an action potential. And there is a stimulus applied, it fires an action potential, tries to fire a second one, and then there's just really not much oomph left in the cell. And that's pretty typical of this kind of neuron. What is different is probably what you're wondering. Well, look here. This is a very subtle difference. Here we have the baseline membrane potential, or what I will call the resting membrane potential. That's that fundamental charge that's put on the neuron in part by the action of the sodium-potassium ATPase. It's almost minus 60 millivolts in the control, 5 millivolts in the AHC patient. So there's an abnormal resting membrane potential, which we would have predicted. There's going to be a less contribution to the resting membrane potential because of that. The other thing that we see is the height is blunted. It's not getting up as high as the other. And that's also something we would have predicted if the sodium gradient isn't as strong and that the amount of sodium that rushes into the cell is less. So these are two things that we noticed. This is a compilation of lots of cells. Those were single cells that we recorded from, but Chris Thompson in the lab, who made these recordings, generated data on nearly 20 cells from each group. And here is just simply if we add in more and more current left to right, you get more and more action potentials. And there's a subtle difference in here. We have to do some more analysis. But on the right is the real major observation, which is this resting membrane potential is less negative in the cells. Again, predicted a little bit based on the loss of the ATP1A3 activity. Now, this was just one pairing of one patient and a control subject. We have another cell line that we got from Kevin's lab from a male patient with the same mutation. And the question is, do we see the same thing? It's a different experiment, different person, but the same mutation. And in fact, we do. Here is the female patient that I just showed you. Here's the male patient. The trend is the same. There's significantly different levels of resting membrane potential. The absolute values are a little different, and that we'll have to sort out whether that's a true difference or not. So there's clearly an abnormality in the resting membrane potential. Another property that's different, and this is a more complicated property, is the amount of electrical current we have to inject into the neuron to get it to fire an action potential. That is called the rheobase current, and it's essentially just us dialing in the volume control on the experiment. And the amount of current that we have to inject is greater in the mutant cells by a little bit. It's still, it didn't in this experiment shake out to be significant, but in another experiment it did. So those are two things that we found that are fundamentally different. Next step is going to be to look at some additional pairings of patient and control, look at some other mutations, and then begin to move on and look at the other types of neurons. For example, we decided we would look at motor neurons. Motor neurons was just an easy way for us to get started. We didn't really expect anything, but we found a difference. Motor neuron potential. So you can see here when we inject a little bit of current, you get a flurry of action potentials in the control subject. But in the mutant line, we don't see anything. There is a resistance to firing. Now, admittedly, we need to repeat this a few more times, but we found this to be a very dramatic difference, and it began to give us thought about, well, why is this? Well, if we look at the resting membrane potential now, which is on the right, instead of being less negative, it's actually more negative. So the motor neurons behave very differently. So I think the only reason I'm showing this is to, number one, illustrate progress. Number two, illustrate there's heterogeneity among neurons. No big surprise. And third, to get the clinicians in the audience thinking about, is there motor neuron dysfunction in this disease that we've sort of overlooked or that's been masked at central nervous system defects? So this is a single motor neuron in culture. It's not in a patient. So anyway, again, a collaboration with Dr. S's lab, we've been able to use neurons from these stem cells derived from AHC patients, and so far clearly have a less negative or depolarized resting membrane potential, very much in line with a loss of function of ATP1A3 in those cells. And we think it'll pan out that those neurons will actually require a stronger stimulus to activate. A curious observation on motor neurons that I think we will follow up when it's time. So anyway, that was the second part of the talk. So now we are about on time. I think the next talk, who started at 1/4 of, we're right about there. And I would recommend we just move forward unless there's maybe a couple burning questions.
I have a burning question. Siggy from Iceland, wondering if, and I don't know if this is something that you have worked on, that there is a GABA deficiency with the that have been with the mutated mice. And GABA deficiency has a lot of symptoms that are very much like, and GABA has to do with the sodium-potassium pump. It doesn't work unless it has GABA. So have we tried to get to see if the mice will work. I know that it's difficult to get GABA through the blood-brain barrier, but still,
well, I think maybe we can talk offline a little bit about what you're citing, but GABA is a neurotransmitter. It's made in neurons. It's released as a chemical transmitter to stimulate the adjacent neuron. The way the neuron amino acid and converts it, it doesn't reach the brain. It's not how it gets there. So giving it to mice would not really have any effect. So again, I think there is almost certainly a role for GABAergic neurons in this disease, which I think there's some evidence from McCarty's mouse model that, but I think there's a lot more to learn. But again, we haven't done a GABA therapeutic experiment.